So a great feature of a theranostic treatment is that we can use a PET scan imaging to select patients that are very likely to benefit from the treatment. This has been shown quite convincingly with lutetium dotatate for neuroendocrine tumours or lutetium PSMA for prostate cancer. So perhaps unlike many other treatments in oncology where we don't have a reliable, let's call it a predictive biomarker for response, and we just treat everyone, here we can do a total body scan and make sure that all the lesions, all the metastases are expressing the target, and hopefully that's the majority of patients, but at least we don't treat the patients that are very unlikely to benefit from treatment. This is a real kind of personalised approach, and maybe now we would even call it precision oncology, and we think this is the way of the future. So gallium-68 is a short half-life positron emitter, and it's unique because it is made in a generator, which is a small device that can sit in a radiopharmacy, and it can make gallium-68 on demand. So it does not require a cyclotron, it does not require a nuclear reactor. It's a device, and it has a shelf life of 6, 9, even 12 months, and there's a variety of manufacturers that are making gallium-68 generators. So this enables on-demand radiotracer production for PET-CT scanning. It's very easy to label to small molecules, peptides, reliably, and it also has a short half-life of 68 minutes, which means patients are exposed to only very small doses of radiation when they have a gallium-68 PET scan. And I think we've seen from both dotatate and PSMA neuroendocrine and prostate cancers that this can become a new standard of care and made very widely available, either from radiopharmaceutical distribution hubs within a city where it's made centrally and then distributed to the hospital, or in larger hospitals within a hospital radiopharmacy department in itself. So broadly speaking, there's two ways we can target a receptor. One is with a small molecule and one is with an antibody. And I'm a big fan of the small molecule approach. This has really, again, revolutionized treatment of neuroendocrine tumors and prostate cancer. And the advantage compared to antibodies is that the uptake is very fast and it's eliminated by the kidneys. And we don't want the radioactive substance circulating in the bloodstream for a very long time, which is what happens with an antibody-based approach. So the antibody-based approach often has a very high incidence of hematotoxicity, lowering of blood counts, and this enables you maybe to give 1 or 2 cycles, but no more. And we want to give multiple cycles of treatment to be effective, and a small molecule approach really enables that. It's also easy to manufacture and distribute, much simpler than an antibody-based approach, and this means that accessibility and availability to patients may be better with a small molecule peptide approach. So one of the great things about PET-CT is that we can quantify the uptake, and we use the SUV or standardized uptake value as a measure of this. So on a FDG PET scan that we use as a general oncology imaging agent, we typically see SUVs in the 5 to 15 range. This means that the uptake in the tumour is roughly 5 to 15 times more than the background activity. And for a PET scan, this is done at 60 minutes. For the theranostic agent, it's a little bit different. Lutetium has a half-life of 7 days. But in order to deliver high doses of radiation to a tumour, the higher the SUV in tumour, the better the tracer will be. And with dotatate and PSMA, which have both been FDA-approved successful theranostics, we can see SUVs frequently over 50, even over 100, and sometimes as high as 200 to 300 at the extreme upper end. But this is really very, very high uptake, and it means that you may be getting 50 to 100 times more radiation to tumours than normal tissues, and this is a very large difference. And at least the early results of this tracer are kind of, let's say, in the PSMA or dotatate range, or maybe even higher, which is quite astounding. And we have a lot of new traces in nuclear medicine. We study a lot in our centre, and we see very, very few that come through that have this kind of extraordinarily high SUVs, which we think will translate to high tumour targeting when we switch from a diagnostic gallium-68 to a therapeutic lutetium-177.